Hey, this is Coach Boydston, and in this screencast, we're going to be speeding it up with enzymes. And so enzymes are going to be something that speeds up a reaction. And if you take a look at this enzyme here on the left, what you're going to notice, it looks vaguely familiar to one of our four main macromolecules. I'll give you a second to guess. Yes, it is a protein. If you said protein, you were correct. So enzymes are proteins, and what they do is they speed up a reaction just to make it a lot faster than it normally would be on its own. And we give these things a name. We call them a catalyst. And I like to have goofy ways of remembering stuff. So one way I remember what a catalyst is, remember, a catalyst is going to be like our enzyme. Enzymes are proteins, but it speeds up reactions. And so I have this couple pictures here. If you look over here in this top right, I have this catapult here. It's old medieval catapult. And obviously this thing is going to take what's sitting right here and it is going to launch it high in the air. Just like if you look down here at the bottom, this catapult, it's already gone through the deal. It was sitting right here and it launched this direction, launching this person's sky high. So like a catapult launches things into the air, all right, a catalyst is going to launch the reaction into hyperspeed. And so that reaction that would normally take place at a certain pace is now going to speed up because that enzyme is present. And again, we call enzymes or catalysts. They speed up the reaction. So to better understand this, we need to know this term activation energy. Activation energy is just simply the energy needed for a reaction to take place. And so to understand how a catalyst works, let's take a look down here at this bottom left graph here. This graph right here represents just a normal reaction without a catalyst, so without an enzyme. And you'll notice we have a reactant here, and then it has to go through a certain amount of energy that it's going to have to release or reach in order for that reaction to take place and to produce our products. So this space from here to here is the energy needed for the reaction to take place. So we would call this level right here the activation energy, the energy that's going to need to be required for that reaction to take place. And you notice it's quite a bit of energy. If we glance over at the right one, and let's look at both of them together, you'll notice this represents a reaction in which we are using an enzyme or a catalyst to speed up the reaction. And one thing you'll notice, we have the same reactant, but you can see here it's much less energy is needed for that reaction to take place and produce our product. And so this is the activation energy here when there's the presence of an enzyme or catalyst, which is much lower than what you have over here to the left. And so how does a catalyst or enzyme speed up a reaction? Well, it lowers the amount of energy needed for that reaction to take place. And so with a lower energy needed, obviously the reaction is going to take place faster. And so that's how it speeds it up. And just to bring up this picture, just to show you, enzymes, like you can see this enzyme here, they have these little active sites. So this little space right here where a substrate will attach. Now a substrate is just anything that an enzyme is acting upon to help speed up a reaction with. And so substrates will attach here, and then we continue on. It's going to form what we call an enzyme substrate complex, and then it's going to produce our products. And so those products will be released from this reaction. Now, one thing I want you to notice about this, there's actually two things, but the first thing is this. You see this enzyme here? It is not changed. So the first thing I want you to know is that enzymes are reusable. So it doesn't take a whole lot of enzyme to act upon a whole lot of substrate. All right, so enzymes are reusable. They don't break down after the reaction and gone. They just move on, they find another substrate, and they react with it. So enzymes are reusable. The next thing I want you to think of here is these active sites. I want you to think of these active sites as the lock. All right, the enzyme's the lock. The substrate is like a key. And so this whole lock and key concept. And what does that mean? It means that enzymes are only going to act upon a particular substrate. So there's not just any substrate here that can enter this lock and actually open that door. All right, so enzymes are specific to the type of substrate that they are going to act upon. So if I have an enzyme here and I have a substrate and it's meant for that one, well, it can act upon it. Now if I come across and I have a different substrate And I got the same enzyme. It can't act upon that one. So it's only specific to a particular type of substrate. So again, enzymes are specific. They're also reusable. Hopefully this will help you understand this whole idea of specific. If you look here, I got a little chart here of these are called 5 food enzyme families here. Just to bring your attention to a couple, like up here you have lipase. Well, guess what that acts upon? Lipids. It's an enzyme that, if you look down here, will act upon oils like this lipid here. So lipase is an enzyme that will help break down oils or lipids. Another one that's really common here is lactase. Now, lactase is an enzyme that is going to act upon lactose, which is a molecule found inside dairy products. So if you are lactose intolerant, then you are lacking, or this enzyme is in very low supply. And so lactase acts only upon lactose, nothing else. So lactase can't act upon a protein. Lactase only acts upon lactose. It's specific. Let's take a closer look. If you look at this diagram in the top left, just to drive this home, lactase is the enzyme, whereas lactose is the substrate that is going to attach. And you'll notice that it has the ability to attach at this active site of lactase. Why? Because lactase as an enzyme is meant for lactose. It's going to connect, and then it's going to produce our products, which are glucose and galactose. And then this enzyme, remember, is reusable, so it's going to continue on. Now, if you look at this example just below it, you have sucrose. Well, lactase is not meant to work upon sucrose. And so in this case, no reaction takes place, and obviously lactase has no ability to break down sucrose, which sucrose is just 2 glucose molecules. And so it doesn't have the ability to do that. So they are specific. And if you look down here at the bottom, here's just a closer look at that same reaction. You have lactose, and you'll notice here it is a disaccharide. Di meaning 2, saccharide, sugar. So it is a carbohydrate. And then with the use of lactase and a little bit of water, we're going to break that down into galactose and glucose during this reaction. So lactase can only act upon lactose. Enzymes are specific. You'll also notice here that we added water to this reaction, which you remember from a previous screencast means that this was a hydrolysis reaction. Total opposite of when we take water out of the reaction, we call that a dehydration synthesis reaction. So if we go back to our diagram here of an enzyme working on a substrate, remember, this substrate is specific to a certain type of enzyme, and it's going to go through the process of producing the products, and then remember, this enzyme is reusable to do this process all over again. All right. Now, the last thing I want to talk about here with enzymes is there's 2 main factors that are going to affect the enzyme function. So meaning how these enzymes will function in our body. The first one here is enzyme, as far as enzymes action is going to be pH. And so if you look at this pH, you'll notice this little dotted line, which shows kind of where the enzyme works the best, is right around 7. Now on our pH scale, 7 is neutral. And if you don't remember pH, I'll bring it up here in just a minute. 7's right in the middle. The pH scale just goes to 14. And so what this chart's showing you is there that this enzyme works best here. Anytime the pH goes down less or gets more, then the enzyme's functioning is getting less and less. So just to refresh you on pH, if we take a look at this chart, you'll notice in the middle you have 7, which is neutral. Anytime we go less than 7, all the way down to 1, we're becoming more and more acidic. And so that's going to be, you can see lemon juice or any type of acid that we would use. Anytime we go higher than 7, it's becoming more and more what we call basic, or on this chart it uses alkaline. So it's becoming more basic. And so right there in the middle at 7 is where we have our neutral pH, where most are going to work the best. An example of this would be these 2 here. In digestion, we have a couple enzymes that work for us, pepsin and trypsin. And if you're looking at this chart, let me ask you a question, see if you can figure it out. Which of those 2, pepsin or trypsin, works best in an acidic environment? Give you a second. If you said pepsin, then you were correct. All right, you'll notice here the optimal working environment for the pepsin enzyme is somewhere right in here around 2.5. which we know 7 is neutral. So anything less than 7 is becoming more and more acidic. So pepsin is actually an enzyme found in our stomach. We know we have stomach acids, and so it's really good that pepsin is an enzyme that works really well in an acidic environment. Trypsin is released by our pancreas, also helps with digestion, but you'll notice here it works best somewhere closer to that neutral mark. All right, so one of the factors that's going to affect how enzyme works or how it functions is going to be the pH of that particular thing. The second thing is going to be temperature. And if you'll look at this chart over here, you'll notice when it's 0 degrees Celsius, we have a little bit of enzyme function, but it's not very good. Think about when you're cold. When your hands are cold, you can barely move those things if they're freezing, if you've been outside in really cold weather for a while. But as that temperature goes up, you'll notice also the enzyme activity goes up, and it reaches its full potential here at 40 degrees Celsius. And then you'll notice as the temperature continues to go up, then that enzyme really falls really quick. What we call that is enzyme denaturing, which means that enzyme is going to denature. So what do you think's happened with enzyme functioning here at 70? Well, there is none. The enzyme is denatured, meaning it's no longer able to work. And so if I pull up this little diagram here for you to see this, when the enzyme is right around 40 or just slightly below, you'll notice here we have our substrate which can attach to this active site and this particular enzyme, and the reaction take place. Now as we go above 40 and that enzyme starts to denature, then what happens is this active site changes. And because it changes, that substrate can no longer attach at that site. It renders that enzyme useless. And so that when an enzyme denatures, then it no longer functions anymore. And that's why if you look back over here on this chart, you'll see that above about 63 degrees Celsius, there's no enzyme activity because that enzyme's denatured. It's no longer able to catalyze or speed up that reaction anymore or help that substrate and speed up that reaction. So just a couple things, temperature and pH that affect that. So this was speeded up with enzymes. And just remember, enzymes are proteins. It's one of our 4 main macromolecules. We call them catalysts because they catapult the reaction into high gear. They speed up the reaction. And then just remember, there's 2 main things about them. They're reusable. They also are specific to the type of thing that their substrate that they're acting upon. And then pH and temperature can affect them. So hopefully that was beneficial to you. You guys have a good day.